So guys, let's move to or continue with our previous topic. Now let's we move towards the or we talk about the second circumvention. We have already circumvented the formation of, the processes which are involved in the formation of phosphoenol pyruvate. Now we want to get, fructose 1,6 biphosphate to be dephosphorylated. Why we want the dephosphorylation of fructose 1,6 biphosphate? Let's we go again to our slide, which is showing us the glycolysis pathway, because that one will be our standard trademark slide. That will always be the reference slide. Whenever you want to study gluconeogenesis, you should have in mind the whole picture of the different pathways which are part of the glycolysis. So here, for instance, you have made phosphoenol pyruvate, fine, and from here until the fructose 1,6 biphosphate is formed, you have reversible reactions, fine, you get it. From fructose 1,6 biphosphate to fructose 6 phosphate, again the reaction is irreversible, fine. This one actually, this one, fructose 1,6 biphosphate to fructose 6 phosphate. So you have to devise a way that this irreversibility, the problem caused by this irreversibility, because we want to move towards the formation of glucose, we have to have some solution. So the nature here, the solution nature had devised here is the invention of another enzyme, fructose 1,6 biphosphatase. As the name shows, it will dephosphorylate the fructose 1,6 biphosphate and change it into fructose 6 phosphate. This enzyme is found in liver and kidney because gluconeogenesis takes place so excessively in liver and also kidney. We have already told you, in normal fasting condition 10% is by kidney and 90% gluconeogenesis takes place in the liver, but in excessive fasting condition it goes to like 40% gluconeogenesis in the kidneys. Then in this way we bypass the irreversible PFK1 reaction, that is phosphofructose kinase 1 reaction. So we will see then how regulation of this reaction actually takes place. So if you remember the fructose 2,6 biphosphate in the glycolysis, we go to that actually slide. You must have gone through it in some of our previous topics, is that fructose 2,6 biphosphate acts as a very potent, perhaps the most potent regulator of glycolysis. So you can think then, if something is so potent, a very highly energetic activator of glycolysis, it will definitely be a very potent inhibitor also of gluconeogenesis, because gluconeogenesis is opposite to glycolysis. In addition, you should also, in addition to fructose 2,6 biphosphate, then the low energy state, if there is if the energy state is low, what will happen? That the gluconeogenesis will also be then inhibited, because there is no much energy already present and gluconeogenesis demands so much energy. But on the other hand, if there is ATP, much excessive ATP present in the cells, what will happen? The gluconeogenesis will be highly active. So ATP act as activator and AMP act as inhibitor of the process of gluconeogenesis. Similarly, fructose 2,6 biphosphate act as the inhibitor of the process of gluconeogenesis. Also, finally, also because this is under allosteric regulation, this enzyme, what is that insulin to glucagon ratio also affect the activity of this enzyme. When for instance, when glucagon is high, then the phosphatase is also very active. When glucagon is low, the phosphatase activity is inhibited. It's no longer so active.